much, Sam, and thank you for the kind invitation. It's a pleasure to be here. So the cardiovascular system has a very important relationship between mechanical force and the biology. So it's not all that well known that the original cardiovascular system at the embryonic stage actually forms in response to force as the heart first starts to beat. I think many of us recognise that during adult stage, the flow of blood and the strain on the blood vessels and in the heart is really important for our normal health. And of course, as we progress to disease stages, such as hypertension, heart failure and stroke, then mechanical forces become really very important indeed, and those diseases are the #1 killer worldwide. What's not been very clear, and is still mysterious in many ways, is how does this interconnection work between mechanical force and the biology? When you look at the literature on this, you see that a lot of people look at force responses after quite a delayed time, so to several minutes after the force has been applied. What we're interested in is that the first event, possibly, or at least one of the earliest and very rapid events, is a calcium signalling event, and we think this is mediated by the piezo-1 channel, so I'm going to show you information relating to that, and I'll be interested to hear your questions. We began this because Adam Patapoutian, who got the Nobel Prize last year, as you've heard during this conference, was studying neuroblastoma cells, and he was hitting those cells, interested in mechanical pain responses and seeing an ionic current. And then they did an siRNA knockdown, and when they did the 74th knockdown, then they found piezo-1 ion channel. What interested us about that was that this channel had a conductance of about 25 picosiemens. Now, we'd been looking for a calcium channel that was the origin of the flow sensing mechanism in endothelium for many years, studying TRPC channels and things like that, but we couldn't find the channel. We were interested in this 25 picosiemen channel because Joachim Hoyer's group had described 7 years earlier that in endothelial cells from umbilical vein, he could see a 25 picosiemen channel. So we thought maybe this is something to do with piezo-1. There was a major problem with that because Adam Patapoutian described piezo-1 as a very fast inactivating channel, which didn't match with what we thought should be there, but I'll come back to that later. And we actually began with HUVEC experiments, and Katsuhiko Muraki, who you heard speak yesterday, did some of the patch clamp studies indicating that piezo-1 is an important channel in endothelial cells. But I'm going to begin here with the mouse studies, because what we thought was that if piezo-1 really is an important flow sensor, then it should be important in embryogenesis, because we know in the mouse that the heart starts to beat around about day 8, and then the blood is pushed into the nascent vasculature. You can actually see that here with this endothelial stain. This is CD31 labeling of these endothelial cells in the yolk sac. Blood is pushed into that, and it's known that the shear stress effect then causes the blood vessels to mature so that the developing organs of the embryo can be provided with blood, nutrients, etc. So we thought that if piezo-1 is important as a flow sensor, then it should be important at exactly that point in embryogenesis, which is around about day 9 and 10, and that's exactly what we found. So in the global knockout, we could still see endothelial cells. They were more disorganized, and what was most striking was we couldn't see maturing vessels. We also saw that at this stage of day 9, but there's no effect earlier at day 8, and day 9, that the embryos were smaller, so there was growth retardation, so even more pronounced at day 10, and then the embryos just atrophied and were reabsorbed. So the global knockout is embryonic lethal. Heterozygote animals do go on to live. There are some abnormalities, but you get normal-ish animals. We also did an endothelial-specific knockout using Tie2 promoter, and we found the same thing, so suggesting that lethality of piezo-1 knockout is due to this vascular effect. We isolated endothelial cells from the embryos, and a nice thing you can do with endothelial cells in culture is you can apply shear stress in a particular direction, and the cells line up in this direction as shown here by phalloidin staining. But with the knockout, so without piezo-1, and then they're disorganized. I'm suggesting to you that what happens early in shear stress sensing is a calcium event. So we measured calcium in the embryonic endothelial cells, we applied shear stress, and we could see calcium elevations, small initially, and then it becomes larger. And as many of you know, with calcium signals, there are all kinds of cascading downstream effects. Those are still not fully understood. It relates to various things, such as ATP release and things like that. In the knockout, there's no such signal, but the VEGF response is relatively normal. Rather nicely, if we studied HEC293 cells and measured calcium, and when they were non-transfected, we couldn't see a shear stress response. But when we transfected in wild-type piezo-1, then we could see nice shear stress responses, suggesting that piezo-1 is a shear stress sensor. So the first evidence that piezo-1 was a sensor of physiological force. The knockout is lethal. So to study the adult stage, and if those are interested in cardiovascular biology, mostly we're working on the adult animal, then we did a conditional knockout using the cadherin-5 promoter and inducing this using tamoxifen. So these animals are allowed to go to the adult stage, and then we conditionally delete piezo-1 in the endothelium. And those mice appear superficially normal, but I'll talk more about that. What we did was then we looked at the mesenteric vessels, which provide blood to the intestines, isolated endothelial cells freshly from the blood vessels. So we're not doing any culture here. And then we're doing patch clamp recording to do outside-out patches, pulled off the endothelium and then applying flow. And what we could see was that there was some constitutive activity of a channel, and we were very interested to see that this was a 25 picosiemen channel because we felt that should be a signature of piezo-1. And then we applied flow, the activity goes up, and then we can inhibit the channels with this non-specific channel inhibitor, gadolinium. And so the conductance was about right, and very nicely, when we looked at the endothelial piezo-1 deleted mice, then we didn't see any channel activity at all. It's completely gone. So that reassured us that we were indeed recording from endothelium, and that these channels depend on piezo-1 and they have the right conductance. We think that these channels are the first, or one of the first things that happens when shear stress is applied to the endothelium, and then might sort of trigger a cascade of events afterwards. So they should be fast responders, and indeed they are. So we've performed patch clamp recordings on these endothelial cells and done various kinds of mechanical stimuli. I showed you already fluid flow, but I'll show you here pressure steps that are applied to stretch the little bit of membrane that's at the tip of the pipette. And hopefully you can see that the channels activate very promptly when the pressure step is applied. They remain active. There seem to be two active channels in the patch here, so first level, second level, around about here, and then they shut off again afterwards. Sometimes in the patches you see sort of ensemble currents because there are lots of channels in the patch, so it looks like a whole cell current, but this is actually a patch of membrane applying pressure pulses. And what I'm trying to illustrate to you here is that there's a very fast response. So this is 50 milliseconds. So within milliseconds you're activating these channels in response to mechanical force, and in the piezo knockout they're not there. And we have nice pressure curves. It's very reliable. For those who don't know about piezo, I'll just give you a little bit of an idea about what the channel looks like. There are cryo-EM structures, not from our laboratory, but started from Rod McKinnon's laboratory, and then Beilong Xiao, and also one from Artem Pataputian's lab. The approach we've been taking is molecular dynamic simulations with Andreas Carli at Leeds. And this seems to work remarkably well, and I think this is very promising in ion channels, as you've seen from some of the other talks, because the cryo-EM structures don't resolve all of the structure, they don't resolve it in full atomic detail, and as other people have indicated, the structures are often in a fixed position, and what you want to see is a dynamic structure. But this is just to give you an idea of what it's like. It's a very large protein, so 286 kilodaltons per piezo-1, and 3 piezo-1s come together, so it's a trimer. In total, around about 900 kilodaltons. There are 114 membrane-spanning domains. I used to think that voltage-gated ion channels were massive, but these are really huge. So in each one, there are 38 membrane-spanning domains, and there are 3 of these together. And it's the last 2 membrane-spanning domains in the C-terminal area that come together in the middle to form what seems to be the ion pore. And above that is a rather large cap structure called a CED, C-terminal extracellular domain. What's quite unusual, but we'll see with time what happens with other ion channels, is what's striking with piezo is that it indents the membrane around about 6 nanometers in its closed state. So the channel probably sits about like this. It's got this cap at the top, and then we think when stretch is applied to the membrane, force is applied, then it flattens and the cap comes up. And quite remarkably, we can see that really quite beautifully in the molecular dynamics simulation, and that was recently confirmed by cryo-EM data, a really beautiful paper in Nature by Beilong Xiao just in April. And the cap comes up, and then the ion pore widens, and that's what we think allows the ions to move through. A little bit crude at the moment, but that's the broad idea of what happens. I mentioned to you earlier on that we were a little bit worried about this piezo idea because the conductance of the channel is right, which is quite nice, but there was a feature of the channels that really wasn't right, and that was inactivation. So there are many piezo papers now describing piezo expressed in hex cells, looking to see what happens, and these are fast activating channels and then fast inactivating, and this is a recording of ours done in hex cells, so we can also see this. It's actually quite variable, but there is definitely a very powerful intrinsic inactivation gate in the piezo 1 channel, so the continued force here, and then the channel decays. This is just a loss of function mutation that we found in relation to disease, and I'm only showing it for technical purposes here to see you don't see anything if you mutate the channel in a disabling way. So the current you see here is through the piezo channel. But as I've shown you already, and I showed you these traces, in the endothelium, the channel activity is sustained, and we've done this in the different configurations, outside out, cell attached, and the whole cell, and I show you here actually a membrane potential recording from endothelium where we apply flow. You get a slight hyperpolarization initially and then a depolarization, as you expect for a non-selective cation channel. And this is completely lost in the piezo knockout. So this sustained depolarization here is due to piezo. And that's really what you want, I think, for a vascular shear stress sensor, is that you can turn it on and then it stays on. And if you increase the flow even more, then you get even more activity. I don't show it here, but you can do nice step-like changes in flow. So that seemed to be a problem, and some people said to us, well, that means that your channel here can't be piezo because the signature is it fast inactivates. So, of course, we were worried by that. We're doing molecular dynamic simulations, and one reason we're doing that, of course, is to see the dynamic states, but we also want to do the simulations in an endothelial membrane. So we know quite a lot about the lipids in the endothelial membrane, and we can put those into the molecular dynamic simulation. And what we noticed was that around the inactivation gate, you can't see the numbers probably well, here 2,476, etc. These amino acids are thought to be the ones that comprise the fast inactivation gate. We saw 2 lipids sitting close to here. These are in the model. One was sphingomyelin, the other one was ceramide, and they're converted from sphingomyelin to ceramide by sphingomyelinases. So this made us think that perhaps there are lipids interacting with the inactivation gate that prevent it from inactivating normally. In fact, there's a very early paper from Sani et al. Suggesting that sphingomyelinases are somehow involved in shear stress responses. So we, I'm just cutting a long story short here, but what we did was we took the native endothelial cells from the mesenteric artery and we put them in the presence of a blocker of sphingomyelinase. It's actually a neutral sphingomyelinase 2 inhibitor, a pharmacological agent here. And what was really beautiful, we found, was that now in the endothelium, in the red trace here, you see that the channels inactivate, just like you see in the overexpression system. So all of a sudden, the endothelial channel becomes like the overexpressed channel. And if we put then ceramide in with the inhibitor of the enzyme, then we go back to the sort of flat, non-inactivating signal, suggesting that the endogenous lipid regulation is controlling the activation state of the channels. Doing it with sphingomyelin or phosphatidylcholine doesn't do anything. We were wondering if we could test this independently and also identify which sphingomyelinase was involved. And the most common sphingomyelinase in the vasculature is neutral sphingomyelinase 2, which is also called sphingomyelinase phosphatidiesterase 3, SMPD3. Very convenient, yeah. And it so happens that there's already a mutant form of this that's produced in mice, actually just loses the catalytic domain. So we collaborated with Natalie Auger's group in Toulouse, and we obtained those mice from her. And we were very, very pleased to see, as you might imagine, that we had a fast inactivating channel, just like you'd expect for piezo, in the endothelium, native channel now. So, basic idea is that when there's no force, the channels are not very active. There's actually some basal activity, which I think is interesting in its own right. But then we apply force, and we see increased activity of the channels, and then we continue the force, and the channels remain active, we think, because ceramide remains high, because there's sphingomyelinase activity. But if we disable the sphingomyelinase enzyme, then the channels do activate initially, but then the inactivation gate comes on and then switches off the channel. So these are real recordings of ours from the endothelium here under the 2 conditions. So what we think is that piezo is the first thing that happens when shear stress is applied. It causes this calcium permeable channel activity, so there's sustained activity in the endothelium. So what might it be doing? So we then wanted to record in vivo what happens to blood pressure. We did that using telemetry. First of all, on the left, if you look at mice that are just fairly inactive, wandering around, measuring their blood pressure, there's not much, actually, there's no significant effect. There might be some effect if we increase the N numbers, but it's not very striking. But what was striking was that if we let them run on a running wheel, because, of course, the thing of piezo is all about sort of physical type things, so make them more physically active, then their blood pressure goes up, just like it does in people during exercise, and that's strongly blunted by the piezo knockout. So it seems that piezo is particularly important in blood pressure control during exercise. And of course, we're very interested in that because exercise is a big thing in cardiovascular health. As you might know, the World Health Organization says that sedentary lifestyle is the 7th biggest killer worldwide. It's supposed to be the sitting down disease. There are other factors, of course. We're all sitting here. You know, we have to do this kind of life. Yeah, it's not very good for us. Yeah. So we're thinking, our idea is that piezo might be a type of exercise sensor that mediates the cardiovascular health benefits of exercise. So again, I'm sort of cutting stories short here. We've just recently published this study in JCI, where we've done slightly longer deletion at the adult stage of the piezo-1, but strictly in the endothelium, to try to mimic what happens if you have a period of a couple of months not exercising. Because we're interested in this, what happens if people go to hospital? Of course, that's common. And then they tend to de-exercise train. It makes them ill. It's quite difficult to recover. It's a common problem. And really, very strikingly here in the endothelium deleted piezo-1 mice here, we see that the activity, so this ambulatory activity, exploratory activity, running wheel activity, is significantly less. And that occurs particularly in the dark phase at night because these are nocturnal animals. We thought this could be because they're less interested in exercise, but that doesn't seem to be the case because they try just as often to have a go on the wheel, but they're just not as good at it. Just like with some people like to exercise, but they're not so good at it. So my son always beats me, but you know, what can I do? I won't go into all the detail, but we've obviously studied this in quite a bit of detail to see what's wrong with these mice. Their muscle actually seems to be normal, but if we look carefully within the muscle, this is actually in the leg muscle, the gastrocnemius, then we find that there are sometimes some collagen sheaths, which is the normal structure that goes around the microvasculature, so that's a collagen sheath here, that are lacking endothelial cells, which is the red thing, in the piezo knockout. So what there is actually microvascular rarefaction, so it's a loss of the microcapillaries. And we think this is due to apoptosis that's occurring in the knockout animal. So the idea is that if you're exercising, then that keeps your microvasculature healthy. If you stop exercising, then there's a rarefaction of the microvasculature in the muscle, and then although the muscle's still there, it's more difficult to exercise the next time you come to exercise. We all probably know that kind of feeling, eh? We've actually worked out the signaling pathway. We think it involves the control of the stability of the eNOS protein in the microvascular capillary endothelial cells, but also a paracrine signaling mechanism involving TSP2, thrombospondin 2, and it's gene controlled by nitric oxide. But anyway, as I say, I'm just sort of brushing through it, but the broad idea is that microvascular piezo-1 we think is very important for maintaining muscle function. And you probably saw a few papers in the literature now about piezo-1 in the skeletal muscle cells themselves. This seems to be a really important thing. So if it's a really important part of the exercise response, then it might be good, not in all members of the general public, you have to be careful what you say about this, but for example in heart failure patients in our hospital who are struggling with exercise tolerance, and that's their big complaint, then maybe it might be possible to stimulate this system to try and help them, because at the moment there is no treatment for that. So we're interested whether it might be possible to stimulate piezo under some conditions, and so I showed you earlier on a flow response in the endothelium. Novartis screened 3.25 million compounds against piezo-1 and piezo-2, and they found a piezo-1 selective activator, and they called it Yoda-1, and there's the structure of it. And if you get into piezo-1, you need to watch all the Star Wars films. We've been making various structural analogues of this. We've got a competitor, we call it Dooku, after Count Dooku, and then there are Jedi compounds as Obi-Wan, and no doubt this is going to proliferate. What's striking is that both of these responses are lost in the knockout. So you can actually quite beautifully reproduce the flow response with this agonist. So we've been making new ones of these because the Yoda-1 is actually quite poorly soluble, has a lot of protein binding, it's very unstable, these kind of things that you get when you're very familiar with that kind of thing. So we've been making analogues. I'm not showing these structures today, but we're hoping to publish this soon. They're actually more potent and reliable than Yoda-1. They have better solubility characteristics and better microsomal stability. One of these analogues here is very effective at relaxing the portal vein. In the portal vein, you actually get a nitric oxide effect. It's completely blocked by L-name, as shown here, and it depends on piezo-1. So, could it be safe to give a person a piezo-1 agonist? Well, I don't know. certainly will not be volunteering yet. We're trying to find out. But we do know that some people have a gain-of-function mutation in their piezo1. This was discovered first in 2012. It was associated originally with very rare cases of anemia. But now it's been discovered that a third of people of African descent have gain-of-function mutations, which seem to protect them against malaria. And it also seems to give them improved physical performance, improved running, jumping performance, things like that. So we've been looking at one of these. This is the human mutation, just one of these. There are quite a few of these. This is a mutation that causes gain of function, and it sits in this outer cap structure. We've been doing this molecular dynamic simulation, so we can also look at we can compare the human and the mouse channel, and they seem to be pretty much identical at this site. So then what we've done is we've mutated this residue in the mouse channel. We've made a mouse that's got this gain-of-function mutation, and really nicely it has the same features as the human disease. So it has this type of anemia. It's called stomatocytosis. These look like coffee bean red blood cells. Show that flow responses in the normal red blood cells, and these can be amplified by this mutation in heterozygote homozygote form here. And what's really striking is there's a lack of recovery. When you stop the flow, they just remain active. We've been thinking, of course, then, what then is going to be the wider consequence? We know this has been identified through studies of anemia, but could there be wider consequences? Well, we described a few years ago that piezo is also in cardiac fibroblasts. So we looked in these mice. So these are the wild-type mouse calcium responses, and then this is the gain of function. And there's a really big increase in the calcium signal in the cardiac fibroblasts in this gain of function mouse. So we're thinking, well, maybe gain of function causes some change in the heart. And indeed, we've looked, and these are still quite young animals, so we're still only partway through these studies. So what we do find is that there's cardiac hypertrophy, increased heart weight, and increased heart weight to body weight ratio. There are other details in the paper we just published this. Increased left ventricular mass and intraventricular septal thickness in systole. We found evidence of fibrosis, increased myocyte size, and increased signaling of things like tenacin C, which is commonly associated with heart failure. So to conclude overall, piezo1 is activated by shear stress. We found that and published that first in 2014, as well as other forces such as increased membrane tension. We think it's a rapid native force detector in response to physiological forces. It's really important in endothelial cells and other cells in the cardiovascular system, but also very broadly, in fact. There's an intrinsic inactivation gate that's disabled in the endothelium, and I think in other cell types commonly as well, such as red blood cells, through a lipid mechanism. The channel is important in blood pressure elevation in exercise. It's important in exercise performance, muscle capillary density. There are opportunities for small molecule agonism, and the gain of function seems to cause a cardiac hypertrophy phenotype. So this has been quite a long project, but we've pretty much enjoyed it so far. And a lot of people have been involved. I'm not going to read through all the names, but I'm hugely grateful to everybody's names. You're probably grateful I'm not going to read through everybody's name, but it doesn't mean I don't appreciate what they've done. It's been fantastic, and we've had really superb funding from these agencies, and thank you for listening.
Thank you, David. Brilliant talk. Very nice. Questions for David?
If it's a Star Wars question, I'm not answering it.
It's not a Star Wars question. Now, I was thinking about the expression of the piezo channels in other parts of the body. So I think when we talked before, I was talking about it being expressed in the GI tract. And I was just wondering whether it might be possible that it might be stimulated as the food passes through the gut in a shear stress way.
It could be, yes. Well, this is not really known, but there are published data on epithelial cells and the importance of piezo1 in those. And it's actually been suggested that piezo1 might be a sensor of short-stranded RNA that's produced by bacteria in the intestine. But in terms of peristalsis and all of those things, I think this is wide open. The channels are there, I think, and yes, I think that'll be interesting.
Lovely talk, David. I was sort of just thinking back to the TRPV4 story, where the TRPV4 knockout shows some sort of similar biology to piezo. So so do you think piezo is sort of upstream of TRPV4?
This is what we're finding increasingly. So lots of pathways have been described to be mechanically sensitive. We've reported recently on one about the notch pathway, known to be mechanically sensitive. We've shown that piezo is essential for that process. So this is also true for TRPV4. I think there's a lack of evidence that TRPV4 itself is directly mechanically sensitive. And also P2X, signaling ATP signaling downstream, connexins and things like that. It does seem to be, and it's not just us saying this, that piezo is a key starting point.
And also, it seems slightly counterintuitive to me anyway, but most things that elevate endothelial cell calcium cause vascular relaxation. So that doesn't seem to be the case here. So what's the explanation?
Well, it's also a good point, and yes, we've published on this, and it does seem to depend on which vessel you're looking at, and I think there's a lot of heterogeneity. But of course, it's a non-selective cation channel, so it causes depolarization, which is opposite to EDHF, endothelial hyperpolarization. So there is potential for a dichotomy here of constriction and dilation. Yeah, so we think in the mesenteric vessels, under certain conditions, like exercise, they're actually mediating constriction, but in other vessels mediating dilation. Because in exercise, the mesenteric vessels constrict, as you know, to move the blood more towards the muscles. We've started to look at the expression pattern across the body. We've got a specially tagged form of endogenous piezo1 we've done by CRISPR, and it does look like there's diversity in how the channel is expressed and how functional it is in different places.
Thank you.
Assuming there are no serious cardiac side effects then with such agonists that you're describing, do you think this could promote longevity? Is it that sort of fluoride in the water that might be used in the future?
As you know, we need to be careful with this kind of thing. But what I think it is true that exercise, physical exercise, does increase the length of high quality life. Not everybody can exercise all the time and things like that. So if you could mimic that in some way. Physical exercise is a complex whole body thing, yeah. But a lot of the vascular health aspects relates to the shear stress sensing mechanism. So yes, possibly yes.
And Yoda did live to quite a good age, I think, as well.
So many thanks for the exciting story. I've got a couple of questions. First of all, it does look like a very fast-acting channel. So do you see similar timescale in your simulation? Like very fast changes happening for the pore opening?
Yes, the challenge with MD simulations is that they're all on very fast time scale. So this is all done in the nanosecond scale. So for an electrophysiologist, that's a bit shocking. But of course, these are real molecular changes that have been simulated here. So yes, it's very quick, actually.
The second question, it may sound very stupid, but do you know whether there has been any association with ceramide content and any hypertension or heart failure or whatever?
Yeah, good question. I don't know, but yes, that could be.
Yeah, thanks. Thank you.
Yes, okay.
Can I ask you a question, David? Are the piezos subject to any kind of receptor-dependent regulation? Can the sensor be tuned, or is it set for life?
There's actually not much done on that, but there is an early report in sensory neurons suggesting that as you activate receptors, muscarinic receptors, etc., then you deplete PIP2, and that does regulate the channel. And in our MD simulations, you see quite a lot of PIP2 interaction, so that almost certainly there's a PIP2 regulation.
Good, interesting. So let's thank David again for an excellent talk. Thank you, David.